In this lesson, we're going to be talking about the properties of eukaryotic cells. We talked a little bit last time about prokaryotic cells, and now we're going to focus on these more complex cells that are eukaryotic, that have all these various parts inside. We're going to be talking about a lot of these different structures. They're called organelles, and we're going to talk about what each is doing in this eukaryotic cell. So eukaryotic cells have these things called organelles. The purpose of organelles is to separate the metabolic reactions that are taking place. So whatever's going on in one area of the cell is not affecting elsewhere in the cell. So if there are enzymes acting, or if there's a difference in pH in one area of the cell, it's not going to affect the rest of the cell. Most of these eukaryotic cell organelles are surrounded by their own membranes, and the organelles that you'll see in a eukaryotic cell will vary depending on the type of cell. Sometimes they'll vary in number, and sometimes they'll vary in type. The first organelle let's talk about is the nucleus. The nucleus of the eukaryotic cell is the site of the DNA, so you're going to find the DNA here in the nucleus. This is an electron micrograph of a nucleus. So the microscopes that we have in class, of course, we can't see in this kind of detail, but this is taken with an electron microscope. The DNA in a eukaryotic cell is wrapped around some proteins that we'll talk about later on in the year, and we call this chromatin. So you can see in this picture some little dark areas, that's the chromatin. The nucleus itself is contained by a double membrane, and it's called the nuclear envelope, and it has these things called nuclear pores, which are little holes within the membrane that regulate what's going in and out of the nucleus. You will also see in most eukaryotic cells a nucleolus, which is a darker area, and this is the site of the production of these things called ribosomes. Now, much of the organelles that we'll talk about are surrounded by membrane, and these various organelles that are membranous actually work together. It's a whole, you can think of it like a factory, and they're all connected to one another. We call this the endomembrane system. It's a whole system of membranes, starting with the nucleus and then ending up with the cell membrane that surrounds the cell. There is a whole series of steps that will take place for many of the products that the cell is manufacturing. This endomembrane system is involved in the synthesis of proteins and the transport of proteins. Sometimes transport is by way of these things called vesicles, which are these little sacs filled with stuff. It includes the nuclear envelope, part of the endomembrane system, the ER or endoplasmic reticulum, the Golgi apparatus, sometimes called the Golgi body, the lysosomes, and then various vacuoles. Let's start with the endoplasmic reticulum. We generally just call it ER for short. It is an extensive network of membranes that is continuous with the nuclear envelope, so you'll always see endoplasmic reticulum attached to the nucleus itself. There are two regions that differ in structure and function. The first region is called the smooth ER, or SER. You'll see it here in this micrograph, and you'll also see it here in this drawn version. The smooth ER is the site of synthesis of lipid. It's a site of metabolism of carbohydrates. It's a place where calcium is stored, and it's also important in detoxification of the cell. And to detoxify, they add an alcohol group, and then that inactivates whatever the molecule is. One thing that I found interesting is that I guess when there's an increase in drug use, I'm talking about maybe even just medicines, something that you take that you need to take, over time, sometimes drugs are less effective, and apparently what happens is that the smooth ER, the cells will increase the amount of smooth ER in order to deal with this medication that you're taking, and the more smooth ER that's there, the more it's inactivated by these detoxifying reactions that take place, and then you have so much smooth ER that the drug really doesn't have any effect. So we call that drug tolerance, and I guess the smooth ER is involved with that, which I find kind of interesting. The rough ER looks very similar in this little cartoon drawing here, and then here in the micrograph. The rough ER is called rough because in these micrographs it appears sort of fuzzy, and it's because it has these attached ribosomes. Ribosomes are the sites of protein synthesis, so the rough ER is involved in the synthesis of protein. specifically secretory proteins. These are proteins that are going to be exported a lot of times out of the cell. Many of these are glycoproteins, which are proteins with an attached carbohydrate. Glyco is referring to carbohydrate. The finished proteins are often secreted out of the cell in these vesicles, and they are always wrapped up in membrane. Let's talk a little bit about the ribosomes themselves. These are found in all cell types. So these are found in prokaryotic and eukaryotic cells. They're the site, like I said, of protein synthesis. Some of them we call free ribosomes. These are the ones that are just floating around in the cytoplasm. Others are attached to the ER, like these here. Free ribosomes produce enzymes that function in the cytoplasm, the cytosol. The bound ribosomes, the ones that are attached to the ER over here, are the ones that produce proteins that are going to be packaged into vesicles. So two different types of proteins produced in the two different types of ribosomes. Next is the Golgi apparatus. This is the shipping and receiving center. It is a set of flattened membrane sacs, sometimes hundreds of these flattened membranes. They receive the proteins in vesicles from the rough ER. They modify the proteins and release them from vesicles out the other side. So you can see here, here are vesicles coming from the rough ER. They enter here at the Golgi body or Golgi apparatus. They are modified throughout these membrane sacs, and then they're secreted out the other side. And then oftentimes this vesicle is going to go up and merge with the cell membrane itself, and then it's secreted out of the cell. Now, oftentimes these vesicles will have protein markers on them, or glycoprotein markers, and these are kind of like zip codes that tell where the vesicle should go. The Golgi apparatus also is involved in the manufacturing of polysaccharides. Now, also part of the endomembrane system are the lysosomes. Lys means to split, so or to break down. These are sacs of hydrolytic enzymes that digest macromolecules. In the lysosome, it's a low pH within the lysosome, and the enzymes that are in the lysosomes need that low pH. And the reason for this, of course, is that if the lysosome bursts open, then the enzyme is not going to digest the whole cell, because at that point the pH will change and the enzyme will be inactivated. So that's kind of ingenious. The lysosomes are produced by the Golgi apparatus. Oftentimes cells will engulf and consume food from outside of them, and now we're talking about maybe single-celled unicellular organisms, not necessarily like your body cells, but they will, this process called phagocytosis, which is how they eat, they gobble something up. Oftentimes they'll create this thing called a food vacuole where the food is contained, and oftentimes these food vacuoles will merge with the lysosomes, and then the cell will be able to digest the food that it's eaten. Lysosomes recycle the cell's material, and the products of that digestion will be made available to the rest of the cell. And I thought it was interesting to know that sometimes there are certain disorders associated with malfunctioning lysosomes. For instance, Tay-Sachs disease, which is a genetic disorder, it results from a lack of lipid-digesting enzymes in the lysosomes. So without having those lipid-digesting enzymes, lipids build up in the cells, and specifically the cells in the brain are affected, and this genetic disorder causes a problem with the brain. So it's kind of interesting. So here's an overview of the endomembrane system. We're starting here with the nucleus. The nuclear membrane is continuous with the rough ER. So proteins that are made here on the ribosomes in the rough ER are packaged up into little vesicles by the ER. These vesicles travel across to the Golgi apparatus or the Golgi body, which is usually not too far away. They're processed within the Golgi body, and then they're packaged up again into a transport vesicle and moved to the plasma membrane where they're secreted out of the cell. Now, sometimes they are not. Sometimes it might become a lysosome, and it's used within the cell, or some of these vesicles might be for substances that are going to be used by the cell and not secreted at all. Now, technically, I do not think that vacuoles are part of the endomembrane system, but they are also sacs that are made of membrane. They have varying functions. I already mentioned food vacuoles, which we see in all cells. Some unicellular organisms have something called a contractile vacuole. which helps them with water balance, and you can see them here. They actually can contract and squeeze water out of the cell if it fills up with too much water. We see large central vacuoles, such as this one in a plant cell, which is a reserve of water, proteins, carbs, and ions. Some central vacuoles in plant cells contain pigments, such as the cells in flower petals, and that gives them their color. And in some plants within the central vacuole, they also contain poisonous compounds, which is a benefit for plants not to be eaten, because animals will avoid them because they have that poisonous substance. Now, once again, not part of the endomembrane system, we have the mitochondria. The mitochondria are the site of cellular respiration. That is the conversion of sugars and fats into usable energy in the form of ATP, which we'll talk about more later, and this process uses oxygen. A mitochondria is contained within its own double membrane, and it has some folds called cristae, and then this in-between space that's called the matrix. What's interesting about mitochondria, in addition to the fact that they provide all the energy for the cell, is that they have their own DNA and they have their own ribosomes. And they are semi-autonomous. They might be able to live on their own to a certain extent away from the actual cell. And there's a theory called endosymbiosis theory, which says that at one time the mitochondria were actually free-living organisms, and they were engulfed by another cell and just ended up becoming part of the cell. It's like endo meaning inside. It's like symbiosis, like maybe mutualism, but it's happening inside of a cell. So that's a theory about how mitochondria came about. Now, plant cells have these things called plastids. So this is not in any animal cells, and it's also not in any prokaryotic cells. But these organelles are very important in plants. There are three types: chloroplasts, amyloplasts, and chromoplasts. Chloroplasts you've probably heard of. They contain chlorophyll, which is the pigment that allows cells to collect sunlight and convert that into sugars. And they are the ones that perform photosynthesis. Chloroplasts also have a double membrane system with flattened membrane sacs within called thylakoids that are stacked into these things called grana, which I always like to say they kind of look like stacks of pancakes to me. The fluid in between we call the stroma. And similar to mitochondria, they have their own DNA and ribosomes, and so the endosymbiotic theory also may hold true for chloroplasts as well. You can see in this cell that it's got a lot of chloroplasts. All these little green circles are chloroplasts. Now, amyloplasts, amylo refers to starch, and these are storage sacs for starch that are found in roots and tubers. Here are some potato cells. And you can see these look like little bubbles. Those are all amyloplasts. They're packed with starch. And then chromoplasts, chromo means color. These are the pigments that give fruit and flowers their colors. And chromoplasts generally are reds, yellows, oranges. These are cells of a tomato, and you can see all the little red specks are the chromoplasts. Cells also have these things called peroxisomes, and these are present in both plant and animal cells. They are bound by a single membrane, but they are not part of the endomembrane system. Some break down fatty acids or help detoxify alcohol. And in the process, the peroxisomes actually produce hydrogen peroxide, and then they also make catalase, which you're familiar with, to break down that hydrogen peroxide. So it's the peroxisomes that make all the bubbles when you put the potato with the hydrogen peroxide. So I was talking kind of fast, but I didn't want the video to go too long, but that's all for this video, and we'll see you next time.